In this video, we're going to talk about how do cells know when to reproduce? How do you heal? So for example, if somebody takes a hammer and hits you in the leg, right, afterwards your cells might be destroyed, you have a bruise, right, it hurts. So after some more time, your bruise heals, right? So how does your cells know when to reproduce and grow more cells? Here's another example, right? Like you get a paper cut, right? And then you're bleeding, but your skin doesn't stay open forever, right? Your skin eventually heals up, right? And so that it looks like there's no cuts, right? So I had many paper cuts before, but do you see any paper cuts now? No, because it healed, right? So how does your body know to do cell division? How does it know to make more cells? So inside, right, your cells might receive messages from other cells telling it, hey, you don't have enough cells here. You should probably make the cell divide, right? And so how is it going to make the cell divide? We're going to go ahead and look at the cell cycle. So in the cell cycle, it's just describing the process of the cell determining whether it needs to divide or not. In the cell cycle, most of the time the cell is in G0. G0 means the cell is not going to grow and it's not going to divide, right? So most of the time you can see my hand is still the same size. It's not growing any larger, right? If you did the cell cycle infinitely, you would have cancer or you would become a giant, right? You would just keep growing forever. So most of the time your cells are in G0. You don't need to grow more cells. But if you do have a cut, right, your cells need to grow, so that's G1. Then S phase is when your cells, they need to duplicate the chromosomes, duplicate the DNA, because every cell has the same exact instructions, and so if it doesn't double all of the instructions, then the cells would not have the correct amount of instructions. Then after you duplicate the instructions, that's S phase, you go to G2, the cell grows again, and then finally, step 3 is M. So you're either doing mitosis. Mitosis is for any body cell to make more cells, or meiosis. Meiosis is for sex cells. So when you need to make more sex cells, you do meiosis instead. And then cytokinesis happens where the cells fully separate, and then the whole cell cycle can start again, where the cell either enters G0 and doesn't divide more, or it's going to be in G1 and resume the cell cycle. So all of these steps, G1, S, G2, all of these steps are interphase, when the cell is just preparing to divide. M phase is when the cell actually divides. So how does the cell know to go through these different stages? Well, one enzyme that's important is called CDK, Cyclin dependent kinase. How do you know it's an enzyme? It ends in ASE, kinase, right? So CDK is an enzyme that helps with the reactions needed for the cells to go through the different phases. Cyclin is a protein that attaches to CDK and helps the CDK become activated. And so when the enzyme's activated, it can help with the reactions to go through the cell cycle. So obviously, when you need to make more cells, you would then make sure that your cell knows to produce more of this enzyme, because this enzyme will help it go through a cell cycle. You will also need your cell to produce cyclin to activate the CDK so you can go through the cell cycle. So what is the process of healing then? Healing is when you do mitosis, when you regrow your cells. So what does mitosis look like? So if we look in this picture, right? Before mitosis, here's a somatic cell. Somatic cell is just any body cell with the full amount of chromosomes, right? And you can see that the somatic cells, they have to double their instructions. So the DNA is duplicated, the chromosomes are duplicated. When are they duplicated? They're duplicated in the S phase. Remember, if we don't double the chromosomes, if we don't double the instructions, then if I just separate it out, it won't have the same amount of instructions. So these duplicated chromosomes are called sister chromatids. So sister chromatids are identical to each other. So it's this X shape, and both parts of this X are going to be the same as each other. Centromere is just that middle part. So if you imagine the X, the middle part of the X would be the centromere. See the middle part of the X. Centromere just means the middle part there that's connecting the two sister chromatids together. So let's look once again. Before S phase, it looks like this, the chromosomes, two chromosomes there. After S phase, right, each chromosome duplicated, and we knew it duplicated because they are an X shape. So once that it's duplicated, right, S phase goes to G2, G2 then goes to mitosis. So here's the actual mitosis steps. First step, what's missing? The nucleus is gone. So prophase, no nucleus. Metaphase, the chromosomes are lining up in the middle. Anaphase, we rip apart the two chromosomes, the two sister chromatids. We rip them apart. So each side has the identical DNA. And then telophase, we build the nucleus again. And finally, cytokinesis is not part of mitosis. Cytokinesis is when the cells fully separated out into two circles. Now look at the picture. In these two cells, do they look like the original? Yes, right? So the original cell had two chromosomes, and each of these two cells also have two chromosomes. So you can see why this S phase is so important. We have double the chromosomes, right, into an X shape, and then make sure that each of the cells get the chromosome. Okay, so that's it. That's how you make more cells.